how do the codons know what amino acid to code for? This wheel right here that looks complicated, but it's really not, determines what amino acid ultimately from the 3 codon reading from the RNA. So this is a dictionary of the genetic code. Let's see here. Yeah. So given an RNA triplet, we have 3 letters. It's going to specify the amino acid that is going to result. What I want to show you guys is over here, it's my pointer, envisioned all over here in this lighter purple are all of my amino acids. I can see serine, leucine, phenylalanine, glycine. Amino acids, you can write the full name like glycine here, or another way to write it, and you guys might see it, is its abbreviation, 3-letter abbreviation, always uppercase with 2 lowercase letters. So GLY is going to be the abbreviation for glycine. GLU is the abbreviation for glutamic acid. So these are all the amino acids in purple. I can appreciate here that amino acids are actually written, some amino acids are written more than once. Over here I have, let's see, a serine that I want to point out. In this area I have a serine, S-E-R. Well, I also have a serine over here. So multiple codons can read for the same amino acids. I do want to point out that we have something here called a start codon. I'm going to teach you guys that AUG, I'll explain what that means in a bit, is the start codon. That is basically going to be the start amino acid for every single protein that gets made. What I'm trying to tell you guys is that you need the amino acid methionine to start coding for a protein. Methionine must exist. If you don't have methionine, that organism is out of luck for making proteins and will not survive. You also have, in addition to your start codon methionine and your amino acids, you can also see in red here something called a stop codon. There are different stop codons. There are 3 actually that I'll show you. 3 stop codons, that is basically going to be the endpoint of making the protein. So you start with methionine and then you add the different amino acids, whichever one is going to constitute that protein, and then it stops to end translation. You must reach one of these 3 codons in order to do that. So let's actually read this wheel together so I can show you guys how you go from a 3 triplet mRNA codon to an amino acid. So here's an example, mRNA codon giving AUG. I want to go ahead and read for that. So I'm going to use the wheel to do this. So we start here in the middle. We always start in the middle. And I actually have either 4 nitrogenous bases to start from. It could either be A, it could either be G, it could either be U. We're working with RNA, so it's not a T, it's a U, or a C. So adenine, guanine, uracil, or cytosine. Because this codon starts with A, I'm going to start at the A, reading from left to right. So there's the A, I'm going to start here. Now, this wheel is broken up into 4 different sections from this point. I'm going to stay in this section. I'm not going to go anywhere else. I'm going to continue on from this area. So I can either go here, G, I can either go here, A, I can either go here, C, or I can either go here, U. It depends what the next letter is, and that's going to show me where to go. The next letter after A is U. So I'm going to go ahead and go there. There's U. Here's my section, and I'm going to stay here. Same idea, where the next letter is G. So from U, I'm going to go to G, and now I have which amino acid it's going to code for. It's actually going to be the start codon. That's methionine, M-E-T. If you guys are wondering if you're going to have to memorize this wheel, you are not. I'm going to give you this wheel. I'm going to give you a codon, very similar to this example here, and the next examples we're going to go over, and you guys are going to tell me which amino acid or stop codon it's coding for. You also are not going to need to know the abbreviation for the amino acid. I'm going to give you all that information as you can see on the wheel. Let's do the next one. Maybe you guys can do it before you hear my explanation, see if you get the same result. So mRNA codon, G, 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 G triplet. So I'll always start here. I'm going to go to G. Next, I'm going to go to G. Notice that I didn't go from this G to say this G over here because I need to stay in this quadrant of the wheel because the first letter was not a U, so I would ignore all these other quadrants. I'm staying over here. So here's G to G, and then to end in G, that's glycine. Appreciate GG, it really didn't matter what that third letter was. It could have been a G, A, C, U, that didn't matter. It's glycine. You would get the same amino acid. Multiple codons code for similar amino acids. Others not so much. mRNA codon, UAA, we start here. UAA is one of the stop codons. That is going to code for a stop codon. That is no amino acid. It is just telling that process of translation to end. And now you have your protein. Because diverse organisms share a common genetic code, so that wheel that I just showed you guys, it's pretty neat that scientists are able to program genes into species to produce a protein from another species that they wouldn't code for otherwise. This is basically genetic engineering and transplanting DNA into these organisms. Here's an example I'm giving you guys of a green fluorescent protein that was taken from jellyfish. So these jellyfish make this themselves. Well, scientists took the gene that made this green fluorescent protein, engineered it into a mouse, and we can see the mouse glowing with that protein. The mouse wouldn't glow otherwise, and the mouse, because of the common genetic code, that wheel right here that I'm showing you guys being similar between organisms, that mouse's translation process knows to code for these amino acids. So it's pretty interesting that we're able to do that.